Dear learners, today we are going to learn what are the pre-formulation technologies. It consists of physical and chemical characterization of the drug molecule, which forms the foundation for new drug application process. I am Vineet Agnihotri, Assistant Professor at Shri Vile Parle Kelwani Mandal's Institute of Pharmacy, Dhule. I welcome you all for this important technical learning. Either your drug is in a R&D lab, in a pilot plant, or at a tech transfer level. Everywhere, pre-formulation is a foundation and starting point of dosage form technology. Come together to discuss what are the important aspects of pre-formulation. Dear students, coming to the important part of pre-formulation study, that is, what is the need of this study into the discovery of drug molecule. So first of all, it is an important aspect of new chemical entity. When you identify there are certain critical parameters which affect the dosage form, there you need to understand what sort of changes you could make so that your formulation will be more effective. Second one, before pre-investigational new drug application process, you need to understand how the drug will be approved on the basis of its potential characteristics related to pharmacodynamic, pharmacokinetic and physicochemical properties. And therefore, during new drug application process also, it is very much important to understand how pre-formulation affects the drug approval process. So at the end, new drug application completely depends upon this important aspect of dosage form technology that is called as pre-formulation. After initial screening of the new chemical entity in relation to in vivo and in vitro studies, we will understand the important characteristics of the pre-formulation, including ionization, partition coefficient, microscopy, salt formation, crystallinity. There are approximately 10 important aspects of pre-formulation that we will understand in this lecture. So first of all, starting with how we are taking a decision to develop a drug. After initial screening of the drug molecule, now you reach to a point where you have to take a decision to develop the drug into a particular dosage form. This decision solely depends upon some of the important characteristics of the drug molecule. For example, ionization pK value will decide the absorption characteristics. Next, partition coefficient will let you know where the drug is more soluble or how the drug will be partitioned into the different type of solvent. Next, in which salt you can form the drug molecule, that is called as salt formation. Crystallinity, that is important for developing the drug molecule either in the amorphous form or the crystalline form. Next, if the drug molecule exists in number of polymorphic forms, so polymorphism you need to understand. Next, the important microscopic features of the drug molecule you have to document. Later on, the intrinsic solubility of the individual drug molecule you will record, and at the end you will understand the particle size, common ion effect and drug stability of the drug molecule. Decision to develop a drug is an important stage in dosage form technology. When we know that new chemical entity after initial screening have a certain parameters, those parameters we will take into the consideration while taking a decision in which dosage form we can formulate this drug. Let's say for example we have chloramphenicol which is available in form A, form B and form C. So form B is more soluble while form A is more stable. So we will select obviously form A due to its stability characteristics. The solubility form in form A of chloramphenicol can be solved by formulating it as a suspension formulation. Because in a suspension, you can add indiffusible solid in a liquid, and therefore your stability criteria can be met with the new drug approval process. Talking about the ionization characteristics of the drug molecule, most of the drugs are available as a acid or base. So strong acid and strong bases are ionized throughout the GI tract. Therefore, whenever this drug molecule comes into the contact with the water, that solution behaves like an electrolyte solution, and hence weakly acidic drug will be ionized at alkaline pH, while weakly basic drugs are ionized at acidic pH. Therefore, we can easily understand ionized part will may be helpful for the solubility of the drug, while unionized portion it is helpful for absorption characteristics of that drug molecule, as unionized portion is more lipid soluble than ionized part. Therefore, there is always an equilibrium will be exist between ionized and unionized portion of the drug molecule. So unionized portion will be easily absorbable, while ionized portion having more solubility. Both solubility and permeability are the important part of any drug molecule to be introduced via GI tract into the systemic circulation. Ionization and dissociation constant can also be estimated with the help of pH partition hypothesis. That is, at any particular pH, the relative concentration of ionized and unionized species in a drug solution can be estimated with the help of Henderson-Hasselbalch equation as given here. After understanding that partition coefficient is the important phenomena for absorption of the drug, N-octanol water partition coefficient is the method which is largely used to understand the distribution of the drug into hydrophilic or hydrophobic solvent, into nonpolar or polar solvent. So therefore, the partition coefficient will let you know what is the amount of drug that will distribute itself into the nonpolar and polar solvent. Nonpolar solvent like a chloroform, benzene, isopropyl myristate or N-octanol, any other nonpolar solvent you can also use. When we talk about the nonpolar solvent and the polar solvent, you have to take into the consideration the hydrogen bonding, donor capacity and inclusion of the drug molecule inside the solvent. Talking about the polar one, you have to understand what is the dielectric constant of that polar solvent. Let's say for example, water has a dielectric constant of 78 approximately, and ethanol have a dielectric constant of 25. If you go towards the non-polar one, the dielectric constant is going to decrease. For example, benzene has a dielectric constant of 4. N-octanol water partition coefficient can be calculated by adding definite amount of the drug into the N-octanol and water mixture, that is 2 immiscible solvent mixture. The drug will distribute itself into both the solvent. After distribution, N-octanol water partition coefficient can be calculated by adding definite amount of the drug into the N-octanol and water mixture, that is 2 immiscible solvent mixture. The drug will distribute itself into both the solvent. After distribution and saturation, you can easily calculate the partition coefficient as per the Nernst distribution law with the formula given here.